dear students. I hope you all have been doing well. Today we are discussing about mechanism of membrane transport, and we would be starting this lecture from a simple cell. We all knows that a cell is surrounded by a plasma membrane, and this plasma membrane is actually a lipid bilayer that is having protein embedded in that. And this membrane is a semi-permeable membrane. So what if I am saying that this membrane is a semi-permeable membrane? So what comes in your mind? So what is mean by semi-permeable membrane? It means that it is allowing certain molecule and ion or substances to cross this membrane and stopping other molecules to cross it. So question is this: why and how this membrane could be act as semi-permeable membrane? To understand it, we have to understand the chemistry of this membrane. So we all know this: the plasma membrane is a phospholipid bilayer. So what if we will look on the chemistry of the phospholipid that is present in the lipid bilayer? We see this: each phospholipid molecule is composed of a polar head that is having phosphate charge. So phosphate charge is a negative charge on it. So due to this phosphate charge, this head, phospholipid head, is a polar site or polar head. However, the other part of this phospholipid is having two hydrophobic tail. And if we look on the chemistry of the protein that is embedded on this plasma membrane, so this protein is all actually having two charges, positive or negative charges. Positive charge is remain outside and the negative charge is remain inside. So due to this, the presence of this phospholipid and the protein in this lipid bilayer, these phospholipid and protein decorated this plasma membrane with the positive and negative charge. So what happened? And here is one more factor, that there are certain protein that are soluble protein that are present inside the inner side of the membrane, like in the cytoplasm. They are also having certain charges on that. So due to all these charges of the protein and the phospholipid, this membrane is decorated with uh, uh, positive and negative charge. So how these charges are making the difference? They are repelling certain ions and the polar substances to come inside the membrane. And similarly, if these are the present, these charges and the substances, these ions or substances are present inside the membrane, so they would be repelled by this membrane from inside the membrane and could not be able to go outside. And one more factor is here that is not allowing to transport substances and ions from inside to outside the membrane, that is the uh, tightly packed nature of these phospholipid molecules. These phospholipid molecules are so tightly packed with each other. So due to this tightly packed nature, this phospholipid molecule stop the entry of certain bigger substances to enter inside the membrane or to go outside the membrane. So what happened then? If these ions and substances keep repelling with this plasma membrane, so what happened? Uh, Cell would not be able to carry on its biochemical processes. Cell must need to have these molecules, substances, and ion to engulf or to take inside it in or to export outside from the cell to regulate the biochemical processes in the cell. So what happened then? How the cell do this all biochemical processes? Cell to do all these biochemical processes and to get to take up certain molecules and ion inside the cell and to export export certain molecules and ion from the outside towards the outside of the membrane. Cell membrane adopts certain biochemical mechanism uh, of that regulate the entry of these molecule and substances inside and outside of the membrane. So today's topic, our topic of today's talk is to understand what are these uh, type of the transport or the mechanism by which cell regulate the entry and the exit of the certain specific molecules and ion from inside and outside of the membrane. So these all our transport mechanism are divided into three basic type. One is the simple diffusion, other is the active transport, and last one is the other transport. These are non-conventional method that are used by the cell to transport uh, to transport certain byproduct or the product that are synthesized in the inside the cell, or to take up certain type of the product that are much bigger inside from bigger in size from outside the membrane to the inside of the membrane. So here is the outline of all these mechanisms that we are going to study today, and we would be studying one by one all these mechanism. And first we would be studying the diffusion. Diffusion is of two types: simple diffusion and facilitated diffusion. Facilitative diffusion again is two types: channel mediated and carrier mediated. Active transport is also two types: primary active transport, secondary active transport. And in the last we would be studying about the mechanism or other mechanism or non-conventional method, and we also say them as a vesicular transport. These are the endocytosis and exocytosis. So from here we would be starting from our first method, and that is very simple method, and we say is the simple diffusion. So what is the simple diffusion? Simple diffusion is actually a passive transport, and it is not requiring any energy, and it is the method of the transport of the small non-charged, non-polar hydrophobic molecules and ions from the area of higher concentration towards the area of lower concentration through the plasma membrane. So what happened in this type of the transport? All those molecules that are not having any charge and they are small in nature, they are hydrophobic, like the oxygen, carbon dioxide, and other certain steroid. They are easily diffused across the plasma membrane from the area of higher concentration and from to the area of lower concentration until and unless the concentration of these molecules get equal on both sides of the membrane. And the this type of the transport, when the molecule uh, moves from higher concentration to the lower concentration, this is actually a law, and we say it's fixed law. This method of diffusion is described by a scientist whose name is Fix. And what happened in this type of transport, there is no ATP required, but question is this: if there is no ATP that is required by this method, so from where the energy comes, and how, what are the driving force of these molecule that is transporting or moving uh, these molecule from higher concentration to the lower concentration? So this driving force comes from the water of hydration. We each knows that molecules like carbon dioxide, oxygen are hydrated, and certain other molecule that are present in the cell, that these are present in the form of uh, combination with water. So to cross the plasma membrane, these molecule must be shed their water molecule from them. Once they sheet the water molecule from them, the sheeting process create an energy. So this energy is a driving force that drive the movement of the molecules, a non-polar molecule, from higher concentration to the lower concentration. And here in this table we can see that there are certain polar substances and they are having some water. Means these are showing that um, this activation energy, that is heating energy, is based on the number of the hydroxyl group. The substances that are having larger number of hydroxyl group having higher activation energy, that is actually the shedding energy that is created when molecule or substance remove water from it. So higher the activation energy, higher would be the transport of the molecule from higher concentration to the lower concentration. And all this phenomena is based on certain factor. So one of the most important factor is the surface area of the membrane. If we are having the larger membrane that is having larger surface area, so the diffusion rate would be higher. If the concentration gradient of the substances and molecule would be higher, so the rate of diffusion would be higher. If the thickness of the membrane would be higher, then the rate of diffusion would be lower. If the molecular weight of the substance would be higher, then the rate of diffusion would be lower. So here we are going to take an example of the simple diffusion. And what is this example? This is the osmosis. So what is this osmosis? Osmosis is actually the transport of water molecule from an area of higher water potential towards the area of lower water potential across a semi-permeable membrane. And in other words, we say that osmosis is the movement of water molecule from an area of low solute concentration to an area of high solute concentration across a plasma semi-permeable membrane. And this movement takes place until and unless the number of water molecule get equal on both sides of the membrane. So one of the best example of osmosis is the hemolysis of red blood cell. So how we can understand this? Just assume that we are having a red blood cell and we are putting this red blood cell in a beaker that is having high solute concentration. So how high solute concentration in a beaker means there is low water potential. When there is low water potential in the beaker and we are putting a red blood cell in that, what does it mean? It means that red blood cell is having high water potential inside the red blood cell. So water will move from inside the red blood cell to the outside, and it result on the shrinkage of the red blood cell. And it happen when we put this red blood cell in a higher solute concentration solution. Now we are thinking about the hypotonic solution. If we would be putting a red blood cell in a hypotonic solution means a solution that is having low solute concentration. So when there is low solute concentration, so it means there would be high water potential in the beaker. So in this case, the water move from outside of the cell to into the inside of the cell, and it results on the um, um, swelling of the red blood cell. And at times will come then water keep moving from outside of the cell to the inside of the red blood cell, and this red blood cell get burst because of the high um, uh, content of water. And there is another example, that is isotonic solution, when the solution would be having equal amount of water and equal number of solute. So there would there would not be any net change of the water transport. The amount of water that would be coming from coming out from the cell would be entering in the cell in equal amount. So now think about that. What is the difference between osmosis and diffusion? So what is the difference? Osmosis always require a semi-permeable membrane. However, diffusion is the movement of a uh, solute or the means the ions from the higher concentration to the lower concentration. And Yeah, osmosis is also the movement of a water molecule from low solute concentration to the high solute concentration. So this was the difference between the osmosis and um, diffusion. Now think about, uh, we have studied about how non-polar, non-charged, hydrophobic small molecule move. They move via diffusion. But think about that what happened with larger polar molecule and ions. How, like water and glucose, how they transport across the plasma membrane. How they transport? They transport via a protein that help the transport of these polar molecules and larger molecules across the plasma membrane. So these protein may be a channel protein or a carrier protein. So the movement of the larger polar molecule across a plasma membrane through channel or carrier protein is termed as facilitated diffusion. And in this diffusion, what happened? The molecule move along the concentration gradient and they do not require any ATP, and they move along the concentration gradient from higher to lower until and unless across the plasma membrane until and unless an equilibrium get reached. So here we are taking an example of the as I said that this facilitated diffusion is of two types, channel mediated and carrier mediated. So we are now taking an example of the channel mediated facilitated diffusion. So channel mediated facilitated diffusion means that there would be a channel present in the plasma membrane that is allowing the entry of the certain molecule to cross or enter in the in the cell from the outside, or allowing the exit of the certain molecule from inside to the outside membrane. So mostly the water is a molecule that transport across the plasma membrane using this channel protein, and this special type the water use a special type of transporter or channel that we say as aquaporin. So what is the mechanism of the water transport through this aquaporin, and how this water molecule could be able to transport means transport across the membrane using this aquaporin channel. So what happened, we can see here a picture of a aquaporin channel, and actually this aquaporin are a protein, and they are having selectivity filter. So what is this selectivity filter? Actually this aquaporin channel is a integral protein that are having certain loop of polypeptide chain that traverse across this plasma membrane, and these polypeptide chain of aquaporin arrange itself across a plasma membrane in such a way that that it arrange its polar or charged amino acid towards its core, and they make a channel. So what happened? Water is a polar molecule. So what happened? This charged amino acid attract the polar water molecule in such a way that they allow entry of this molecule in a single file from outside of the cell membrane to the inside of the cell membrane. So here we are having another example of channel, for example, ligand gated channel, mechanically gated channel, always some certain channel like sodium potassium that remain always open, and voltage gated channel. So the mechanically gated channel are open uh, under the influence of certain stimuli, and the leaky channel like the sodium potassium leaky channel, they remain always open, and they help neuron to maintain their resting membrane potential. However, the voltage-gated ion channel open under the influence of certain means threshold of voltage, like when membrane voltage become minus 50 MV, so they become open. And ligand-gated ion channel open under the when a ligand bind on with this channel. For example, here we are taking an example of acetylcholine. Acetylcholine is a ligand, and this is the acetylcholine channel. When this acetylcholine bind with its binding site, so it allows to open this channel, and they allow the movement of ions across to travel from outside to the inside of the membrane. So now we would be discussing about carrier mediated facilitated diffusion. So what is the carrier mediated facilitated diffusion? As the name is indicating here, it is using a protein that is taking part as a carrier, means it is carries a substance and then releasing from one side and then releasing to the other side. So here we are taking the example of a glucose transporter. Um, so we see these are glucose transporter are present in the small intestine. We can see that this glucose bind on the one side of this glucose transporter, and as this glucose bind one side, there would be a conformational change, and due to this conformational change, what happened that this glucose transporter release this glucose to the other side of the membrane. And as this glucose transporter like as an enzyme, so they obeys Michaelis Menten saturation kinetics. So what is the Michaelis Menten saturation kinetics? It shows that these transporter behave like an enzyme, and what happened uh, when we see that what is the enzyme, when if we are having an enzyme, so it is having certain active site. So here, for example, here is an active site, here is an active site, here is in one active site, other active site, and other active site. So what would happen, the ligand bind with active site, and then until all the active site remain occupied. So as the and substrate bind with the active site, uh, um, the velocity get increased. However, when all the active site become occupied, although there would be extra substrate, but they would not bind with any active site, and the velocity get constant. Why velocity get constant? It shows that all the substrate that was present have bind with the active site. So this is the Michaelis Menten saturation kinetics. So now we will see what is the we have understand that we have understand the channel mediated facilitated diffusion and the carrier mediated facilitated diffusion. So what do you understand there is the difference between channel and carrier mediated facilitated diffusion? In the channel mediated diffusion, they require a channel protein, and in the carrier mediated facilitated diffusion, they require a carrier protein. And what is the difference between simple diffusion and facilitated diffusion? Simple diffusion does not require any channel or the carrier protein. However, facilitated diffusion require a carrier or a channel protein to transport molecule from one side of the membrane to the other side of the membrane. Now we are moving towards discussing the active transport. So what is the active transport? Active transport is a mechanism of the transportation of the certain ions, molecules and the substance from an area of a lower concentration towards the area of higher concentration utilizing ATP across the membrane. So uh, yeah, and this is the non-equilibrium kinetics. And non-equilibrium kinetics we can say because there is no requirement, or when the ions move from the area of lower concentration towards the area of higher concentration, there would be no equilibrium that maintain among the concentration of the molecule. What happened in this type of transport, that just uh, an enzyme help to transport certain molecules and ions from area of lower concentration toward the area of higher concentration. And what does this enzyme do? This is what does this enzyme do? Actually, this enzyme utilize ATP, and how this utilize? Once what happened, that substrate bind with this enzyme, and this ATP bind to the inner side of the enzyme, and what happened, this ATP hydrolyze into ADP and generate phosphate, PI. So this PI bind to the other side of the enzyme, and there would be a conformational change that open up the means this hole of this enzyme and allow the entry of the molecule to cross this enzyme from the to cross this plasma membrane from the lower concentration to higher concentration. So sodium potassium ATPase is one of the example, or sodium potassium ATPase or sodium potassium pump is one of the example of this active transport. And how the sodium potassium ATPase work? It actually it pumps 3 sodium outside from inside to outside, and pumps 2 potassium from outside to inside uh, after the hydrolysis of 1 ATP. And all these sodium potassium ATPase are frequently expressed in the cardiac tissues. And here are certain type of active transport, that is uniport. When there is the movement of molecule in only one direction, we say that this is the uniport. And there is another type of active transport, that is co-transport. It is symport or antiport. Symport is the movement of molecule, two molecule towards same direction, either from outside to inside or inside to outside. Or antipode is the movement of two molecules, one moves from outside to inside and other move from inside to outside. We have talked about the sodium potassium ATPase, and now we would be and all these are the primary active transport. Now we would be discussing about the secondary active transport. What happen in the secondary active transport? In secondary active transport, there would be another substance that would be moving with a primary with the transport of that would be moving with the transport of the certain other chemicals, ions. So what happened here? We are taking an example of sodium potassium ATPase and the glucose transport. So we can see here through the sodium potassium ATPase, uh, 3 sodium is going out and 2 potassium coming in. When 3 sodium going out from inside membrane to the outside membrane, so there would be higher charges sodium that will be present in the outside of the membrane, that create a potential gradient. So due to this gradient, what happened? This gradient facilitate the entry or the transportation of another substance, that is glucose from outside of the membrane to the inside of the membrane. So here we would be discussing about the summary. We have discussed about the diffusion. We have discussed about the simple diffusion, facilitated diffusion. All these were the passive transport, and we have discussed about the active transport, that is require ATP in the form of energy. So here we are discussing another example of the active transport, that is calcium ATPase pump. And so this pump is allow to release the calcium from endoplasmic reticulum, and it utilize ATP in the form of for the means release of energy. Another example of active transport is potassium ATPase. This proton pump is present in the gastric tissues, and we have discussed this thing that the secondary transport may be symport, uniport, antiport, and symport is when two molecule moving together. And one of the other example of the active transport is sodium glucose transporter. These also require energy for the movement of the glucose, and side by side this is an also example of this symport, because once one sodium molecule go inside of the cell, it allows the entry of one glucose or a galactose with it. And now we would be discussing about other ways of membrane transport. So other ways are the endocytosis and the exocytosis. Endocytosis are divided into three type of other transport, that is phagocytosis, pinocytosis, and receptor mediated endocytosis. So yeah, so for the if we compare these two mechanism, means the passive transport or the active transport, we see that passive transport does not require any energy. However, the active transport require the energy, and the active transport is the uphill from lower to higher, and the passive transport is the downhill from higher to lower. So here we would be discussing about the endocytosis and its type. So endocytosis are having three types: phagocytosis, pinocytosis, receptor mediated endocytosis. So phagocytosis is actually the this phenomena is adopted by the immune cell, that are phagocyte, for example neutrophil and macrophages. So what happened in this, uh, a bacterial particle are engulfed by a neutrophil or macrophages, and then there would be a lysosome that fuse with the phagosome. It make a phagolysosome, and the lysosome is a having hydrolytic enzyme that destroy the bacteria inside the phagolysosome, and then this bacterial product released from the these phagolysosome. And the pinocytosis is the cell drinking. And then what happened, that certain substances are present outside the cell that are dissolved in the water. So cell take up the plasma membrane take up this water dissolved substances via process of pinocytosis, via creating certain vacuoles. Then these dissolved material release in the cytoplasm. And the third one is the receptor mediated endocytosis. In the receptor mediated endocytosis, what happened? There are certain type of receptor that express on the membrane. These receptor take up the ligand. So for example, we are taking here an example of the LDL receptor. So LDL receptor are expressed on the plasma membrane, bind with the LDL molecule. Then what happened in the inner side of the membrane? Certain other protein expressed that we say as clarithin. This clarithin allow the plasma membrane to invaginate and they take up these receptor bounded with ligand towards inside of the membrane via making a vacuole. And after taking this vacuole inside deep to the cytoplasm, there are the some molecule that proton that come inside in this vacuole, and then they do lysis of this means break the bond between this receptor and the ligand, and after that the ligand dissolve in the plasma membrane and the receptor express again on the plasma membrane. So here is the exocytosis. Exocytosis is the mechanism by which the protein that synthesized inside the cell transport outside the cell via certain vacuoles that transport from the Golgi towards the outside of the membrane. So what happened as we know? So as we know, in the nucleus, we are having DNA. DNA transcribe into the RNA via the process of transcription, and this RNA transport from the nucleus pore into the cytoplasm, where they come and bind with the ribosome that are present on the endoplasmic reticulum and synthesize the protein. Once protein gets synthesized, this synthesized protein come into the first modified inside the endoplasmic reticulum, then export to the Golgi apparatus, where that certain type of modification takes place in this protein, and then there are certain vesicles that bud out from this Golgi apparatus, and these budded vacuoles having modified protein. These modified protein transport through these vacuoles towards the plasma membrane and then export outside the cell. So this is all about the transport mechanism, and I hope you like this lecture. Keep watching my videos for other lecture. Thank you very much.